Good morning. Thank you for joining us for today's webinar at the New York Botanical Garden. I'm Lawrence Kelly, Director of Graduate Studies at the Garden, and it's my pleasure to welcome you for today's presentation, Evolution and Development of the Seed Coat in Gymnosperms. At any time during today's presentation, please feel welcome to submit questions by clicking the Q&A button at the bottom of your window, and we'll raise these questions for our speaker to answer at the end of the presentation. Live captions can be enabled by clicking the CC button, then show subtitles or view full transcript. Today's presentation is being recorded and will be available in the NYBG lecture library. You're joining us for a most celebrated event, the PhD defense of our outstanding graduate student, Cecilia Zumajo. At the New York Botanical Garden, we love our graduate students. And we treasure the university partnerships that allow us to fulfill this important part of our mission. Cecilia is enrolled through the Graduate Center of the City University of New York PhD program in biology. She has written a PhD thesis based on her 5-year research project, and today she fulfills the CUNY requirement of presenting a public seminar on the subject of her dissertation. Cecilia will give her presentation, answer your questions, then she will go directly into a 2-hour defense with her thesis committee. Special thanks to the members of the committee for their involvement in Cecilia's research project. Now I'm happy to turn it over to Barbara Ambrose, Director of Laboratory Research and Associate Curator of Plant Genomics at NYBG. Barbara is Cecilia's thesis advisor, and she'll introduce the presentation.
Thank you, Larry, and good morning, everybody. It's too bad we can't all be together to go through this milestone of Cecilia's PhD defense, but I have to say, I think this is probably one of our most well-attended PhD defense seminars. Cecilia got her bachelor's degree from the University of Antioquia in Medellin, Colombia in 2015. And she first came to the Garden in the summer of 2016 on an NSF Eden undergraduate fellowship to do research on fruit development. She then started her PhD in August of 2016. She has 9 publications, 6 of which are first authored peer-reviewed publications. She's attended a lot of conferences and courses. Cecilia is always eager to learn more. She's attended some of these conferences and courses across the United States, as well as in Italy and China. And Cecilia has taught throughout her PhD research at the City University of New York, particularly at the Lehman College campus. And she's a really great teacher and an amazing mentor and role model for young female scientists. And she has mentored 2 amazing undergraduates during her time here, Ellie Mendelson from Brandeis University and Dominique Groffman from Columbia University. So it is my distinct pleasure to introduce Cecilia Zumajo. And I would like to point out that she has received several awards for her presentations as posters or as talks. She received the Bill Dahl Award, Graduate Student Award in 2019, and the Catherine Esau Award in 2020. And I think you'll see by her presentation today why it is that she has these outstanding awards. Please welcome Cecilia.
Thank you, Barbara, for that very kind introduction. I will share my screen and would you please confirm, Barbara, that you're seeing my first slide?
Yes, I see it.
All right. Well, good morning, everyone. And first of all, thank you all for being here, for accompanying me on this very important moment of a journey through the seed, particularly focused on the seed coat. So the seed constitutes a turning point in land plant evolution, and it's an important synapomorphy of all seed plants. However, there are major differences among them. So to approach the understanding of these enigmatic and diverse structures, rich in morphologies, it is necessary to get an insight into the genes that are involved in its development. And that is what this research is about, focused on 5 different species of gymnosperms: Ginkgo biloba, Gnetum gnemon, Taxus baccata, Ephedra californica, and Ephedra antisyphilitica. My thesis and this presentation begins with an exploration into what is known about the evolution of the seed code for the first chapter. In chapter 2, I focus on the expression patterns of candidate genes in Ginkgo and Gnetum. Chapters 3 to 5 aim to have a better understanding of the putative genes that may be involved in the development of the different seed code structures, like the integument of Ginkgo, the bracts of Ephedra, and the aryl of Taxus. We will conclude with an overall discussion and the major conclusions of this work. All right, so let's imagine for a moment a world without seeds. For that, we would have to go back around 400 million years to the early Devonian, when Earth was extremely humid, colonized by ferns and lycophytes like club mosses, and in fact, these giant lycophytes that don't exist nowadays. But if we take a closer look at those seedless plants and their reproductive structures, it is possible to start differentiating some key steps in the evolution of the seed. For instance, heterospores, which is the production of 2 kinds of spores. So the megasporangia will give rise to female gametes, and the microsporangia will give rise to male gametes. Endosporic development, which is the development of the gametophyte inside that spore, and the integument, which ultimately is the key novelty in the evolution of the seed, as it ensures the protection of the embryo and tolerance to desiccation for long periods of time. However, the evolution of the integument is not very clear yet. So let's now take a look at the 3 main hypotheses that have been proposed to explain the origin of the integument and of the seed itself. So based on the aryl of Taxus here popping out, the novel hypothesis suggests that the integument evolved as a new structure. The telome origin hypothesis is based on fossil record, which suggests that the integument is the result of fusion of telomes around a single fertile synangium. And the synangial origin hypothesis is based mostly on living lineages of ferns and the pollen cones in cycas. And this hypothesis suggests that the integument is the result of sterilization and fusion of those sporangia around a single fertile sporangium. The fossil record may also support this hypothesis, as those telomes may have been fertile at some point. So these 2 hypotheses are not necessarily mutually exclusive. So let's now define what a seed is. Here on the right, we have an example of a seed of Ephedra, and if we make a longitudinal section through that seed, it's possible to see 3 main tissues: the embryo, the nutritive tissue, which is going to nourish that embryo, and the protective layer, the seed coat. But the seed is just the mature stage of the ovule. When the ovule gets fertilized, it becomes the seed. Here on the left, we have the corresponding ovule, and if we make a longitudinal section through that ovule, it's possible to see a megasporangium or nucellus covered by the integument. The integument will undergo a series of ontogenetic transformations, becoming the seed coat. Then the ovule is defined as a megasporangium covered by the integument. Since the ovule first evolved around 300 million years ago, it became the most salient feature of all seed plants. But there are some striking differences among the major lineages of seed plants. For instance, most gymnosperms have only 1 integument, where the integument leaves a small opening known as the micropyle, and that opening allows the pollen to enter and fertilize the egg. In most gymnosperms, the micropyle is located opposite to the stalk or the funiculus of the ovule. On the other hand, most angiosperms have two integuments covering the nucellus, where the outer integument shows asymmetric growth, making the ovule to bend on itself and leaving the micropyle located next to the funiculus. But these are the major ovule morphologies for these lineages. However, there are a lot of variation in morphology and anatomy within each clade. For instance, in Gymnosperms, there are many associated structures with the ovule for which the homology is still unclear. For instance, in Natalis, there are these extra structures covering the ovules that many people consider integuments. So, but what are the genes involved in ovule development? Well, many genes have been identified to be involved in ovule and integument development in the model species Arabidopsis thaliana, and we're seeing here a picture of those young ovules in Arabidopsis, and next to it is a drawing of one of those ovules, which I am going to use to illustrate with different colors where each of these genes has a function. For instance, integumenta here in green is involved in the proper development of both integuments. At the bottom here, we have the mutant. So how does the ovule look like when that gene is not present? In this case, the integumenta mutant doesn't have integuments. BEL1 is also involved in the proper development of both integuments. The mutant doesn't have integument. Instead, it has this fleshy structure at the base of the ovule. Inner no outer is specific to the outer integument. The mutant only has one inner integument. Aberrant testa shape, which is also known as canadiform, it's involved in the proper separation of both integuments. So the mutant has one fused integument. All right, so once the integuments have been initiated, integument growth seems to be a matter more of cell elongation rather than cell division. And there are many genes involved in this process as well, like short integuments 1, which is involved in the proper elongation of both integuments, and unicorn, which is involved in the cell growth of the outer integument. The mutant here has a overgrowth. So these are the main genes that have been characterized for integument development. However, the story gets a little bit more complicated because each of these genes belongs to a different gene family. So we know all of this for the mother species, Arabidopsis thaliana, which evolved around 6 million years ago. Now the question is, what are these genes doing in ovules with a different morphology and that evolved much earlier in an evolutionary time? So we started this project with 3 main questions. The first one is, what can the molecular genetics of seed development tell us about the evolution of the seed in Gymnosperms? We also wanted to know if the molecular analysis could help us to better interpret those accessory structures that are found in some Gymnosperms, like in Natalis. And also if we could gather enough molecular evidence to refine one of those hypotheses about the origin of the seed. So to do that, the first thing was to know if these genes are even present in Gymnosperms. So we know from previous studies that inner no outer is angiosperm specific, so it's not present in Gymnosperms. And I look for all the other genes. I did a blast search, in an initial blast search using the Arabidopsis sequence as query to find homologs across all seed plants using different genome and transcriptome databases available. The sequences were aligned, and then a maximum likelihood topology was obtained for each of them. So at the end, I had between 100 and 800 sequences for each of these gene lineages. So I know it's going to be very difficult to see what's happening here. So I'm going to summarize it with these triangles. In purple, we're seeing the angiosperm clades. In blue are Gymnosperm clades. The outgroup here is a green bar. And the yellow stars are pointing to places where duplication events were identified. For instance, in Integumenta, 3 duplication events specific to angiosperms were identified. So The evolutionary history of these genes is very complex. They all have undergone multiple independent duplication events, and many of those duplications are gymnosperm specific, with the only exception of short integuments 1, which is highly conserved across all seed plants. This work was published in 2020. But we have to keep in mind that every time a gene duplicates, many things can happen. So here I am illustrating with this multi-purpose camping tool, a single copy ancestral gene. So one scenario is that when this gene duplicates, the 2 new copies retain the exact same functions, so they are redundant. Another scenario is that the ancestral functions are distributed between the 2 new copies. This is known as subfunctionalization. Or it may be that one of the new copies retains all the functions and the other one acquires completely new functions. So given the complex evolutionary history of these lineages and the multiple independent duplication events, it is very difficult to extrapolate what we know from Arabidopsis to any other species. So now the question is, what is the role of these genes in gymnosperms? So to answer this question, I focused on 2 different species, Ginkgo biloba and Gnetum gnemon. I collected the ovules and seeds throughout their development to describe the morphology and anatomy of this structure. But I was also able to perform expression analysis, and that is the experiment you're seeing here in this video. So when you want to know the function of a gene, the best thing to do is to silence that gene and see what happens when that gene is not present. But functional studies are not possible in a species like gymnosperms that are big trees and have very long life cycles. So being able to perform this technique known as in situ hybridization was key to make better hypotheses on the putative function of these genes, as it allows us to pinpoint exactly when and where that gene is present. For instance, here we have a section of a Ginkgo ovule, and in purple, we're seeing the expression of that gene at that specific stage. All right, so let's take a closer look at the morphology and anatomy development of Ginkgo. And here I want to highlight 4 stages, stage 1, 4, 7, and 8. So during stage 1, the integument starts developing. That integument will grow, overtopping the nocellus, and by stage 4, 3 different regions will be easily identified in the integument: the endotesta, sclerotesta, and sarcotesta. The sarcotesta is the outermost layer, and it's the one that becomes fleshy, giving that fleshiness characteristic to the Ginkgo seeds. It is rich in tannins and mucilage cavities. By stage 8, the seed has fallen off the tree through 2 possible abscission zones, one at the base of the funiculus and the other one at the base or the proximal region of the ovule, which is here stained with red. There is another very important characteristic of most gymnosperm ovules that I really want to highlight, and is that at the base of the ovules, the integument and the nocellus are completely fused to each other. So it's very difficult to differentiate between these 2 regions. The integument only becomes free in the apical portion of the ovule. So that region where we cannot really differentiate between the nocellus and the integument is known as the pachychalaza. All right, so for Gnetum, the story is completely different. It has a very unique ovule morphology. The ovules can grow on, or they grow on these strobili that can be unisexual or bisexual. So here in the first picture on the top left, we're seeing a bisexual cone with ovules growing on top of the pollen cones or the microsporangia from the same node. The first section at the bottom is a section through one of those bisexual cones. And we have the ovules here on top of the pollen cones. Here the pollen is viable, so the ovule is not viable. Another key characteristic is that a Gnetum has extra structures covering those ovules. So for instance, here we have 3 additional structures. The inner one is the integument, which actually corresponds to it's the integument which is going to form the microbile. And there are 2 additional structures covering that integument. When the ovule grows on a bisexual cone, the integument or the inner layer, it's going to be the integument, and there is only 1 extra structure covering the entire ovule. All right, so let's now take a look at the expression patterns of all of those genes that I mentioned before in this species. Sorry. So I know there are many genes, so I'm going to leave here on the top left corner the expression patterns for these genes at 2 different ovule stages in Arabidopsis, and in a different color, I'm going to show you the expression of those genes. So for instance, here Wuschel is expressed in the nucellus, but it also acts in integument development. So I was able to perform this study in the different ovule stages, as well as in the pollen cones and the leaves of Ginkgo. We obtained for the expression analysis of Wuschel, we saw that Wuschel in Ginkgo is expressed throughout the ovule and also in the pollen cone and in the leaves. Integumenta in Ginkgo is expressed at the base of the ovule, so the cell layers that later are going to form the abscission zone. It is also expressed in the apical region of the nucellus, which is known as the pollen chamber, and in the megaspore mother cell. We saw expression also in the pollen grains and in the leaves. For Gnetum, the expression of A. integumenta is in the nucellus and in the integument. Well, let's remember that all of these genes are involved in integument development in Arabidopsis. So BEL1 in Ginkgo, there are 2 copies, 2 homologs, and they both have very similar expression patterns. And in fact, they are similarly expressed to what we just saw for Integumenta. BEL1 is expressed at the base of the ovules and the abscission zone. We also saw expression in the pollen chamber and in the megaspore mother cell. The expression of BEL in Gnetum is in the nucellus. Canary is a gene that is involved in the planar identity of those integuments, that it gives that flat identity of the integuments. But in Ginkgo, we saw expression in the nucellus, and later it becomes expressed throughout the ovule and in the pollen cones and the leaves. For Gnetum, there are 2 copies of Canary with different expression patterns. One homolog is expressed in the nucellus and the integument, as well as in the megaspore mother cell. And the other Canary copy is expressed restricted to the megaspore mother cell and to the apical region of the integument. Unicorn in Ginkgo is restricted to the apical region of the young integuments, and it's also found in the pollen grains. There are 2 copies of unicorn in Gnetum with different expression patterns. One is expressed in the nucellus, and the other one is restricted to the megaspore mother cell and to the apical region of the integument. All right, so from this part, we can make 5 major conclusions. The first one is based on the evolution of these gene lineages, which is very complex, and they all have undergone multiple independent duplication events. Also here we have the expression patterns that we have available so far for Arabidopsis. Gnetum, this is a drawing of a strobilus with the pollen cones at the bottom and the ovule on top, and 2 different ovular stages of Ginkgo, and this is a drawing of a pollen cone in Ginkgo. So with the first glimpse at the expression patterns for this species, it's possible to conclude that the function of these genes in integument development is not conserved, because for most of these genes, we didn't see expression in the integuments in this species. However, we're trying to put together a really big puzzle, trying to put together the molecular data that we have with our understanding of the morphology and anatomy of these structures. So it may be that the differences in the expression patterns that we're seeing are actually due to major morphological differences between these structures. So here we have the Arabidopsis ovules, which have 3 different regions in their proximal distal axis. So they have the funiculus, which is going to hold the ovule, the chalasal region, which is the region that gives rise to the integuments, and the nucellus, which is the distal portion of the ovule. So Wuschel, which is a meristematic gene, is expressed in the nucellus. And in fact, the function of BEL1 in integument development is because it is a chalasal gene. So it's one of the genes that represses Wuschel towards the nucellus, and it maintains the nucellus-chalasal boundary, which is very important for the proper development of the integument. But then we look at Gymnosperms, here represented with Ginkgo, and we have this region where we cannot really differentiate between the integument and the nucellus, known as the pachychalasa. And in fact, we saw expression of Wuschel throughout all that region. But also, if we take a closer look at the youngest stages, the expression pattern of BEL, we see that BEL is expressed few cell layers below where the expression of Wuschel ends. So it may be that the main function of these genes is actually being maintained, but we're seeing major differences in their expression patterns because of the major morphological differences between these structures. Well, another conclusion is based on the fact that we saw expression of all of these genes in the megagametophite. So we can conclude that these genes were co-opted from a sporangia developmental network, but this must have happened much earlier in an evolutionary time, because for some of these genes that have been characterized in mosses, ferns, and lycophytes, it has been shown that they are also involved in sporangia development, which also suggests that the integuments are the result of a sterilization of sporangia, supporting the synangial hypothesis for the origin of the seed, which is really exciting. All right, our final conclusion from this part is specific to Gnetum, and we saw expression of some of these genes in the integument in Gnetum, but we never saw expression in the outer envelopes of these genes. So there is this ongoing debate on whether or not those envelopes are integuments or not. So many morphological and anatomical studies recently have suggested that they are not integuments, and that's why we're calling them envelopes. And our expression results of these integument genes, which we didn't find in these envelopes, suggest also that they are not integuments. So, but I still want to be very clear that we cannot make homology conclusions based on the expression patterns of these genes because of their complex evolutionary history. So I definitely think that more studies are still required in Gnetum, specifically focused on the development of those extra structures. All right, so now we wanted to know if we could identify new genes involved in seed development in Gymnosperms that we may be missing from the model species Arabidopsis. So we performed transcriptomic studies in different seed code structures for these species, and that's what I'm going to show you now. So focused on the integument of Ginkgo, I dissected the different tissues, the young ovule, the integument, the megagametophite, and the collar, which is a structure unique to Ginkgo. I also dissected the pollen cones and the leaves. Everything was sequenced by triplicate. Now I'm going to show you the pipeline I used for the transcriptome assembly, so the bioinformatics analysis, and this is going to be the same from chapters 3 to 5, so I'm going to mention it here, and it's divided in 3 main steps. The first step is the transcriptome assembly step, which starts with the quality control of those over 40 million reads with a fast QC. Based on that quality control, I removed the low quality bases and possible adapters from the sequencing with Trimomatic. I'm going to leave here the wall time. So for how long was that program actually running? Then I perform an assembly for each of the samples with 2 different softwares just to test the best possibly assembly method. I used Abyss and I used a Trinity. And here we have the quality of the assembly per sample, and in orange is Trinity, which is slightly better than Abyss, so I continue working with that one. I perform a reference transcriptome de novo, also with Trinity, and I looked at the quality of that reference transcriptome by quantifying the E90, N50, and with the Basco. All right, so the second step is the annotation step. So the first thing is that I wanted to know if there were any contaminants in my transcriptome. So I used Diamond to annotate my transcriptome against bacteria and fungi that may be associated with soil or with plants, and I removed those sequences. Then I found the open reading frames with transdecoder, and I annotated the transcriptome for gene ontology terms using a BLAST2Go, which also has access to the InterProScan database. The third step is the differential expression analysis. So for this step, all the reads are mapped against the reference transcriptome with bow tie, and the abundance of those transcripts is measured in transcripts per million with RSEM. Then the differential expression levels between those transcripts is quantified by EBSeq. And I filtered those genes according to their statistical significance, so the p-value, and also with the fold change that leaves only genes that have a large change, so lower than -2 and greater than 2. And I further annotated those differential expressed genes with a BLAST2Go again. And all the data analysis and the visuals that you're going to see here were done with Python. All right, so taking an overall view of the samples in Ginkgo, with the principal component analysis, we see that the megagametophite and the integument are the most dissimilar samples. And this was corroborated with a hierarchical cluster analysis, which puts together the integument and the megagametophite in one cluster and all the other samples in another cluster. All right, so the first thing I did is that I looked for those candidate genes that we talked before, and I looked for their expression levels, and I performed this heat map that you're seeing here on the left. So in blue are upregulated genes, and in yellow or whitish, according to your screen, you're gonna see downregulated genes. So it turns out that these genes are really not differentially expressed, and integumenta is downregulated actually. But all of these genes, or this analysis, was done before any of the filtering steps. So before filtering the data for the significance with the p-value and for the fold change, because after those filters were applied, these genes were not retained. But I did find 2,137 differential expressed genes, which you're seeing here on this cluster map on the right. Again, here blue is upregulated and yellow are downregulated genes. So these are too many genes. There are over 2,000 differential expressed genes. So I further annotated these genes, and I decided to focus my attention on transcription factors, because transcription factors are well known to play key roles in developmental processes, because they have this DNA binding activity. So from those over 2,000 differential expressed genes, 134 are transcription factors. And that's what we're seeing here on the left in this cluster map. So I was able to identify homologs of very well-known transcription factors families like TCPs and Apetala2s. And these transcripts definitely deserve attention and a further characterization for their functioning in integument development. But there was one that really caught my attention, and that is Fantastic Four. Isn't that a really cool name? Anyways, so it turns out that Fantastic Four represses Wuschel. So I don't know if you remember Wuschel from the gene candidate approach. Wuschel is a meristematic gene and is expressed in the apical, in the shoot apical meristem. And Fantastic Four, or FAF, is also expressed in that same region. It turns out that there are 4 Fantastic Fours in Arabidopsis, of course, and when they overexpressed those Fantastic Fours, you see restriction of Wuschel expression. So FAF represses Wuschel. But that's all what we know about this gene so far. So what we had to do first is to know if this copy that we're obtaining here was an actual Fantastic Four homologue. So to know that, I did a maximum likelihood analysis for this gene family, and it turns out that yes, it is a homologue, but I was also able to identify several duplication events in angiosperms, which here are in this dark purple. And I identified that the 4 Fantastic Four in Arabidopsis are the result of duplication events specific to Brassicaceae. And there is one duplication event before the diversification of all seed plants, of all angiosperms, I'm sorry. So the Gymnosperm sequences are in fact pre-duplication genes. So I looked at the expression of a FAF in Ginkgo, and on top we have the ovules. And if we take a closer look inside this circle, we don't see expression, and this is the nocellus. The expression is only in the integument. So this supports that Fantastic Four is restricted to the integument. There is no expression in the pollen cones or in the leaves. All right, so from this part, we can make 2 major conclusions. The first one is that the transcriptome analysis also corroborated that the candidate genes are not differentially expressed in the integument of Ginkgo. And the second is that now we're proposing new candidate genes that may be involved in integument development, like Fantastic Four. And we didn't know about this gene from the model species, because here we have the expression browser of one of those copies in Arabidopsis, and we see that it's highly expressed and is restricted to the shoot apical meristem, because FAF represses Wuschel in the meristem. But if we take a closer look at the expression of these 2 genes in the ovules in Ginkgo, well, they overlap expression in the integument, but not in the nocellus. So it may be that these genes are also interacting in the integument of Ginkgo. All right, so now, given the extraordinary diversity of seed coat morphologies, we wanted to know what other genes may be involved in different seed coat developmental morphologies. Like in ephedra is a really good genus to look at this characteristic. I focused on Ephedra californica and Ephedra antisyphilitica. I went to the field to collect these species with the help of this amazing NYBG alumna. And I dissected the young cones, the shoots, the pollen cones, the ovules, and the bracts. And I mentioned that this is a really good genus to assess this question, because in ephedra, there are these extra envelopes, which are represented here in reddish. And those structures vary in morphology. So for instance, in Ephedra californica, we're calling them here bracts. They remain dry as the seed matures. But in Ephedra antisyphilitica, they become fleshy, which is very interesting. So with the principal component analysis, I saw that the ovule, the shoot, and the pollen cones are the most dissimilar samples. And in fact, they make one cluster with the hierarchical cluster analysis. And bracts and young cones are in another cluster. I'm sorry. All right, so for Ephedra antisyphilitica, we saw different patterns for the samples, where the ovule is the most dissimilar sample. And in fact, it is in its own cluster and all the other samples are in another cluster, and in fact, they are very similar to each other. All right, so from the differential expression analysis for Ephedra californica, represented here with this cluster map on the left, I identified 407 differential expressed genes, and from those, 23 are transcription factors. Again, I identified very well-known transcription factors families like MIBs and histones, and it turns out there is something else that I want to clarify here. That is that when I collected the young cone, I collected the ovules and the young bracts together. And then the sample for bracts, it's an older stage of those bracts. So if we make a comparison between the genes that are expressed in those two samples, we can find the shared genes that are possibly involved throughout a bract development. So I found 26 shared genes, and among those, there is an AGL6 homolog, which is well known for being involved in ovule development. All right, for Ephedra antisyphilitica, I identified 524 differential expressed genes, and from those, 34 are transcription factors. So again, I performed the comparison and I identified 48 shared genes. And from those, there are many members of the ethylene responsive transcription factor family, which is a family that is very well known for the function in fruit ripening. And that is very interesting, given that this is the species with the fleshy bracts. I also identified a Matchbox homolog. It's possible to be a TT16 homolog, which is also involved in ovule development. All right, so from this part, we can make three main conclusions. The first one is that there are major differences in the expressed genes between these two species, which makes sense given their morphological differences. Also, we're now able to provide a list of candidate genes that may be involved in the seed code development. And also, Ephedra is a genus that has been proposed multiple times as a possible model species for genetic studies in Gymnosperms because it is very small and has relatively fast life cycle. So providing this transcriptome analysis is the basis for developing Ephedra as a new model species in Gymnosperms. All right, so now focusing on the aryl of Taxus, the aryl is also an extra structure that covers the ovules in a Taxus. It develops from the funiculus, and we see it here in pink, and it grows covering the entire ovule. It becomes fleshy. So I dissected the ovules, the young aryls, the pollen cones, and the leaves for Taxus. Again, everything was sequenced by triplicate. With the principal component analysis, we see a very scattered pattern. The hierarchical cluster allows us to see better the similarities between the samples. So we see here that the green aryl or the young aryl is the most dissimilar sample, and all the other samples are in another cluster. So I identified 788 differential expressed genes in the aryl of Taxus, and from those, 73 are transcription factors. Interestingly, I identified several isoforms of Apetala2. Apetala2 is a gene that is well known to be involved in morphological novelties across land plant evolution. And it's also known to control developmental phase transitions. So it would be really interesting to see what the function of Apetala2 is in aryl development. So because the aryl is also a vegetative structure, I performed the exact same analysis, but for the leaf in Taxus. So that's what we're seeing here, the differential expressed transcription factors in the leaf of Taxus. And when we make the comparison between the leaf and the aryl, in fact, we do see 155 shared genes, but there are over 600 unique genes for each tissue. So that accounts for their morphological and genetic uniqueness. So from this part, we can make the major conclusion, and is that the aryl is in fact a very unique structure morphologically and also genetically. And also we're setting up the framework for future studies characterizing the function of these genes in aryl development, and also if they are conserved across different species that also have an aryl, like in angiosperms. So from this work, we can make several conclusions. And the first one is that the genetic network, as we know it for angiosperms, it may not be conserved across all seed plants. But we also saw that the protein sequences of these homologs are highly conserved. So those shifts in the expression patterns may not be due to changes in the coding sequence, but in the regulatory regions of these proteins. But also we have to keep in mind that there are major morphological differences even among angiosperms. So for instance, the way the placenta develops, the placenta is the region from which the ovules develop. That varies a lot within the angiosperm species. So for instance, in Arabidopsis, that meristematic activity of the placenta gets turned on in the inner layers of the carpel once the carpel is already well developed. But in Petunia, the meristematic activity is always active. So there are definitely changes in the regulation of this genetic network, and mainly those genes that are involved since very early in ovule development. But we also saw that there are major morphological differences between these structures. So we definitely need more studies in gymnosperms. And we also need different type of studies like protein-protein interactions to see if the interactions of these genes are being maintained or not across seed plants. I want to reiterate also that our expression study supports and provides evidence supporting the synangial origin for the seed. And well, the seed is definitely a very diverse structure anatomically, as we can see here on top, illustrated on this row, but also morphologically, developmentally, and genetically. Here we have the evolution of seed plants, including fossil lineages in gray. And it turns out that paleontologists have proposed multiple times independent origins for the integument. So the homology of the seed is a discussion, and the structures associated with the seed is a discussion that we definitely need to be having and that we need to address, even more when the species that we looked at have different transcription factors differentially expressed in these structures. So I don't have a transversal conclusion for the entire work, because these species are all so different. So this study also highlights the importance of working with different species across all land plants, because given the extraordinary diversity, it is very difficult to extrapolate what we know from one species to any other. All right, so I would like to thank all my committee members for their patience, their mentoring all these years. It's been an amazing journey, thanks to you all. And I would like to thank also Rachel and Alejandra for their help with the field work, and my mentors, Fabio and Ricardo, all my friends and colleagues in Colombia who helped me with the bioinformatic analysis a lot, and to all the NYBG curators and the staff who have helped me even since before I got to the PhD program, the plant science students and the EUB students, our undergraduate students who are not undergraduate anymore, and of course to my friends, family, and my mom. I also received support from the Botanical Society of America and the Torrey Botanical Society, and all the computer analysis were done with the National Center for Genome Analysis using their clusters at Indiana University. And I'm happy to take questions.
Terrific. Thank you so much, Cecilia, for that fascinating presentation. It's now time for the public Q&A. So audience members, please go ahead and add your questions by clicking the Q&A button at the bottom of your Zoom screen. And you're also invited to upvote questions that others have already submitted. So Cecilia, a question that came up, how did you choose these particular species for your analyses? And why did you choose them as opposed to perhaps species that are more common and abundant? Was there a particular reason you picked these?
Yeah, so I decided to focus first on gymnosperms because of their key phylogenetic position as the first extant lineage with seeds. And then these species, there are many reasons. So Ginkgo also has a key phylogenetic position, is the only representative of one order. So I am covering one order out of the 5 in seed plants. It also, I actually chose it because of the key morphology of the ovules. Ginkgo is a species that hasn't changed much in the morphology since its first evolved around 300 million years ago. So it's a considered fossil lineage. So that is a key, a bridge, because fossil studies, we cannot really do any molecular analysis with fossils. So this was like a very interesting link to assess those questions about the origin and the evolution of these species. And I also tried to find the species with the different morphologies, like the outliers, to see the differences in the expression of those genes in those weird species.
Excellent, thank you. I think you alluded this a little bit at the end, but if you were to guess what kind of patterns you'd see in other gymnosperms, could you tell us a little bit based on what you've learned from these species?
That's a very good question. I think definitely the expression patterns or the genes involved in seed development are going to be different from those in angiosperms. But I think we definitely need more studies in conifers, for instance, because the ovule morphology in a conifer is very different to what I studied. So it would be really interesting to see. I'm thinking that it's going to be a little bit different.
Terrific. Larry, I think you asked a question in the chat, but I'll pose this to Cecilia. You said such great diversity of duplicates in gymnosperms. Can you speculate as to why this is the case? Could it have practical implications?
Well, the genomes in gymnosperms are way much larger than the genomes in angiosperms. So that may be one of the reasons why we always see a lot more genes in gymnosperms than in the other species. And implications, yes, there are many. So in each of these clades, so the species, the genes that we looked at, the expression patterns, only cover few of those clades, because some of these lineages have 5 gymnosperm specific clades, and we only looked at 2 possibly. So we know the function in those gymnosperm clades, but we don't know the other clades, what they are doing. And there are many possibilities. They can have different functions, or they can have the same function. Yeah.
Terrific, thank you. This might be a little outside, but did you see in your background research similar variation in ovule development in angiosperms that you found through your research in gymnosperms? And do you think what you're finding can also inform studies in angiosperm ovule development? Yes.
Yeah, so for instance, the genes that we are identifying from gymnosperms, many of them haven't been characterized in angiosperms. So we definitely don't know if they are involved in ovule development. They may be also involved in ovule development in angiosperms. And there is a lot of morphological and anatomical diversity in angiosperms. Is even greater than what we see in gymnosperms. There are variations in the orientation of the ovule. There is variation in the number of integuments. So morphologically, angiosperms are also very diverse in their ovule structures.
Terrific, thank you. I'll just mention that you've had tremendous applause in the chat. Lots of people are commenting on how wonderful your presentation has been. I just want to make sure you're aware. People are very thrilled and appreciative of your work. So this is a question from Lewis. Do you think chromatin immunoprecipitation sequencing, or ChIP-seq, to identify binding sites and genes controlled by the TFs would be a helpful future direction, or would you take another future direction?
I think that would open many endeavors. I think that would be a good tool to use in this species. And we definitely need also different tools. We need more genomes available. We definitely need more studies like genetic tools to be standardized in this species, like ChIP-seq. Yeah, definitely.
Here's another question from Larry. What is the evidence that the extra envelopes of ephedra are not integuments? Did you know this beforehand? Do your data make this case a lot stronger?
I did know, like I knew both sides, and so the evidence morphologically, the envelopes develop in a different way as to what happens in angiosperms. So angiosperms have two integuments. The inner one develops first and the outer one develops next. In Natalis, the outer envelope develops first, and then the nocellus with the integument develops. So it goes the opposite way. Also, the anatomy of those envelopes looks a lot more like a leafy structure than an integument. So that's another reason why by anatomy it hasn't been considered an integument. And yes, definitely the transcription factors that we found there are very different to what we know for integument development. So I do think my transcriptome data could support that hypothesis that these are not integuments.
Terrific. A person asks, did you include cycads, or why did you not include cycads in your research project?
Yes, that's a really good question. That's actually a little side project. So I have been working on cycads for the anatomy and morphology, but not for genetics, because cycads have they are very difficult to collect. So if you want to get a really young cone in a cycad, you have to kill the entire plant. And then the cone is all at the same stage, all the ovules at the same stage. If you collect one cone, you get everything at the same stage. So I haven't done it for a more molecular analysis, but we have been looking at the development of the ovules in different cycads with Dennis, who has been collecting cycads for a really long time. So yes.
Terrific, thank you. We're all out of questions from the audience, but I have a question for you, Cecilia. What is next? What are your plans for the future?
Yes, well, I am moving to a postdoc, so I will be working on a postdoctoral position, also in seed development, but now it will be in the model species, Arabidopsis, so I will be using different tools. And yes, it's going to be in Italy too, so that's it.
That's so exciting.
Yeah.
Well, congratulations, and thank you so much. Again, our audience is really excited for you. There's so much wonderful noise there, and you'll get a chance to read that later. So thank you so much for sharing with us and giving us this terrific presentation and summary of all your work at the Garden.
Thank you so much. And thank you all again for being here. Thanks.
Wonderful. So again, to our audience, we're so happy that you came today. If you missed any part of the event or want to watch it again, get into those figures in more detail. A recording has been saved and it will be published by the end of next week on the NYBG lecture library. And we hope you'll join us again for future science, conservation, and humanities lectures with the New York Botanical Garden next week on April 22nd on a Thursday. we'll be celebrating Earth Day with a premiere of a new documentary on the conservation of plants, people, and culture in the Pacific Islands of Vanuatu. And this screening will be followed by a conversation with project collaborators from Vanuatu and from the New York Botanical Garden. So for those who haven't already registered for our mailing list, you can go ahead and sign up by the link I'm about to publish in the chat in just a second. So until next time, please stay well, stay green, and take care. Goodbye, everyone. And thank you once again, Cecilia.